Thanks a lot, and thanks for the introduction and for, so to say, inviting me to present here. So my name is Engkvist. I'm based in Gothenburg, Sweden, heading the molecular AI team at AstraZeneca. Today I will give an introduction what we are up to within AI and automation for drug design. So first to give a general introduction, so what we think so to say AI and machine learning can impact in the drug so to say discovery and development phases. And I think we, like many other companies, are applying AI machine learning end to end from early drug discovery all the way into production. And in discovery and development, 5 typical use cases are in the slide. To the left is target identification. Say, how could you, for instance, use a knowledge graph together with the genomics data, either from GBAS or functional genomic studies, to further identify and validate targets. Drug design, which I'll come back to. Toxicology, how can we do better prediction there, particularly so we can reduce the number of animals needed in toxicology studies. Imaging, which I think is a quick win. How can you basically reduce the time that you need to analyze your images, and also apply it in clinical. So let's say one example there is how can you use natural language processing to quicker identify a patient cohort for your clinical trials. But today I will focus on drug design, and my team is working completely in that area. Drug design can basically be described as 2 things. In a drug discovery project, you have synthesized a set of compounds and analyze them, and then you have the question what to make next and how to make it. And the whole drug design is to answer those 2 questions. And they might sound simple, but of course in a real-world drug discovery project, they are extremely complex. There are a lot of variables and a lot of uncertainty in the data, for instance. So going further, so how do we optimize compounds in the pharmaceutical sector? And usually we have something called the design, make, test, analyze cycle. And in there, you start with hit identification to the left. So you do a screen, so you test a lot of compounds for your target of interest, and then you find a hit. And typical techniques are high throughput screening, DNA encoded library screening, fragment-based screening, but also virtual screening. But that will give you a starting point. And then you do what's called multiple of the DMTA cycle, design, make, test, analyze. And then after, usually we're talking about years here, you come out, you have let's say candidate drug that fulfills your target profile. And it can take, say, around 3 years in average. The variation is very large depending on a lot of factors, tractability of the targets, the in vivo models, and so on. But it takes a long time. And of course, the challenge for us is how can we speed this up? How can we use AI, machine learning, and automation to significantly reduce the time it takes to go from an initial hit over to a candidate drug? And how we are approaching it is basically we try to improve both how we make each cycle as information rich as possible. How can we learn as much as possible in each cycle? And then the second, how can we increase the speed of each DMTA cycle? How can we select the most efficient route? How can we basically make sure the reaction's working? How do we organize the logistics with respect to screening and so on to really be able to get feedback as soon as possible? So we approach from 2 ways, both to try to maximize the learning in each cycle and also increase the speed of each cycle. And the priorities areas, so to say, to the left, you see, so to say, how we approach the problem. And the first part is drive the science. We have the ambition to be part of the scientific community, to contribute with scientific articles, to train the next generation of PhD students, so on, and be We're simply part of the scientific community that drives this exciting field forward. We're also building platforms, and we're building platforms. I don't mean build platforms from scratch. It's more to take established open source tools like PyTorch, TensorFlow, scikit-learn, are the key, and integrate them into a platform. But we also can plug in commercial software if needed, if they are considered to be better. So basically an open source platform, we then also can plug in commercial components. And then of course we have to the bottom left impacted project portfolio. So when you come from a pharma perspective, it's not only to impact one individual project, it's really how can we use machine learning and AI to impact the whole portfolio, a really big part of the projects in all of disease areas and sites. To the right is a few of the most important machine learning and AI areas where we're working. First one is deep learning based de novo molecular design, which I will focus quite a bit on the talk on. So there we're using mainly recurrent neural network, but we and also others have been successful with other architecture like variational autoencoder or adversarial network. And we are doing a lot of new development there. So Jose and Mercado from the team have a poster. So if you've not looked at the post yet, I encourage you to go and have a look at it. How we also not only use string-based method there, but also moving into use graph-based methods. Another example of development is another recent paper where we use molecular transformer in the optimization of compounds. I will not talk much about synthesis prediction. So Espen in the team gave a presentation not so long ago at the RSC meeting, but it's of course another area where we are active in and have also moved forward very quickly in the last few years. We also work with more accurate property prediction. We have worked on that for a very long time. I would say there have been some more evolutionary progress there in the last year by introduction of the graph convolutional network and so on. What has really had impact, particularly for affinity prediction in the last year, has been free energy perturbation, that you can actually do physics-based modeling at a scale now with the GPUs that you couldn't do before. And also another way to improve the accuracy is actually to privacy preserve machine learning. And in the end, I've discussed a consortia with 10 pharmaceutical companies that try to improve models through privacy preserving machine learning. And then to the bottom, of course, the goal is not only to do machine learning, but really go into more AI. So to say, for instance, together with the robotics, how can we go do more autonomous decision making under uncertainty? So here's some examples of science, some publications. One highlight is to the upper left. It was a collaboration with Marvin Seglov, and he was at the University of Münster. And that was actually the first publication that used recurrent neural network for de novo design. To the left, the middle publication, it was the first reInvent publication. And at that time, reInvent was the first code that was open source for molecular design. Now, of course, there's a lot of codes out there for different various architecture. On the bottom are two review articles that are actually 2 years old now, but I think they still can give a good introduction to the subject. And then to the upper right is a very recent article in Nature Machine Intelligence, so we further developed the recurrent neural network. On the bottom is the link to our GitHub page, where we both have the reInvent platform and the AI Syn platform as open source. If people would like to try out it, please go to our GitHub, and there is instructions and an example how we can use our codes. And then if we go to, so to say, the little bit, take a step back, say, go a bit more detail in neural networks and deep learning. So neural networks have been existing for decades. And so if you see to the upper right, it's standard architecture with an input layer, an output layer, and a hidden layer. And then you have a set of descriptors and some experimental data, say solubility. You train your hidden layer and then you can use that for prediction. That existed for quite a long time. It was popular around 20 years ago, but then somehow replaced, I would say, by random forest and support vector machine. And then I would say maybe 2012 or something like that, neural network came back, and then in the deep learning shape. And to the right to the middle, it's a feed-forward network. That's common for several type of predictions, like chemogenomics prediction, and is suitable architecture for privacy-preserving machine learning. And then there's convolutional neural network for image processing. There's autoencoded neural network. And to the bottom right is the architecture that we have used the most, the recurrent neural network, which, as you can see, not only had information transformation from the input layer to the hidden layer to output layer, but also you have information transfer in the side direction from the one hidden unit to the next. And that makes RNNs very powerful for strings, for time-dependent phenomena and so on. Of course, we were very interested, can we use that in drug design and cheminformatics? And why are we interested in it? And of course, as a cheminformatician and drug designer, we would really like to sample the whole chemical space. If you do brute force enumeration, then you can maybe enumerate 10 billion compounds or something like that. That's extremely small part of the full chemical space. There have been methods earlier than deep learning, say genetic algorithms. They could generate a lot of molecules, but they never really looked any good and any really synthetic, accessible, and so on. So we're thinking, how can actually deep learning help out here? So you can really sample from the whole chemical space, which is particularly important if you would like to do scaffold hopping. So you will go from one part of the chemical space to another part. So say looking at recurrent neural network, what they've been very good at in training a lot has been natural language generation. And that's something we use, every one of us, with our mobile telephones. So you train the recurrent neural network on a corpora, and then you have basically you can use that corpora, that trained network, to then predict the next word when you type on a keyboard. And in fact, that's something we could actually transfer to cheminformatics. So basically, if you train a corpora, you have a sentence, say the grass is, then it's much more likely that the grass is green, that the grass is purple. This is so-called a conditional probability. Given a set of words in a sentence, there's a probability distribution what the next word would be. Could you transfer that to chemistry? So say if you have a carbon, and a double bond, what is then the next atom? And then it should be very likely that's an oxygen, because acetaldehyde is a very common compound. And in cheminformatics, we are very fortunate that we have a very efficient language. It's called SMILES. So this is an example of molecules, first like a graph, which is a normal intuitive way of describing molecules, but it can also describe as a string. And you can see through the color coding that each part of the molecule will correspond to each part of the string. And that really gives us a handle, because now we can describe molecules according to a grammar SMILES, which means that we can train a recurrent neural network on a, so to say, a prior of SMILES strings, so to say, for instance, Campbell, if you would like to generate drug-like molecules, another database if you say want to generate natural products or fragments. So then when you have trained an RNN, so you sample it to generate new molecules. And the key concept here is the conditional probability. After the training, you have a probability distribution for each token in your SMILES strings. And here's illustrated to the left the sample probabilities for the first token in the string If it's more green, it's higher probability. And then you sample from that distribution, so you not necessarily takes the most probable all the time. And we can see now which is sample. And first now it sampled and took the oxygen, which was the second most common character, the capital O, which corresponds to an oxygen. And then you will see to the right that from a red O, and I will say when I go through the conditional probabilities to the left, I will draw a molecule to the right. So you can see both the conditional probabilities and the actual molecule. So then if you select first as an O and oxygen, what is then the probability for the next character? And that is heavily biased to an equal sign, which is basically correspondent to most likely a carbonyl group. So that's, it's not 100% that it needs to be an equal sign to correspond to double bond, but it's very probable. And we can see what it selects, and it indeed selects a double bond. And then we will see now how it continuously builds up through the conditional probability, one token after the other. And you can see to the right that it corresponds to a perfectly drug-like molecule. And I think that, so to say, RNN then gives us a tool to sample the whole chemical space. And I think now it's 2020, it's I think fairly acceptable, and a lot of people accept that it's actually a very useful way to generate the molecular ID. I think actually 2017 when we started it, it was kind of a more controversial concept, but it's, I think seeing is believing. You need to do it yourself. You need to train on RNN well, and then you can actually see how it generates very drug-like molecules. And then that solves for drug discovery project half the problem. And now we know how we can sample the chemical space. And we also published several articles in a collaboration with Jean-Louis Raymond in Bern to actually benchmark our RNNs with graph-based hardcore enumeration methods to show that we can actually sample the whole chemical space. You'll find those articles in Journal of Cheminformatics. So now we know how to sample chemical space, but then we need to, so to say, hone in to the chemical space of interest. And we do that through what's called reinforcement learning. Particularly, we apply that we would like to scaffold hopping. If you would like to optimize a series, you can also use transfer learning. That works perfectly well as well. But what we do in reinforcement learning, we have like an in silico mini design, make, test, analyze cycle. So we generate molecules and then we basically score them. And then through reinforcement learning, we adapt the weights in the recurrent neural network to generate more molecules that are highly scored. And that's in practice an efficient way of generating molecules. Of course, one needs to make sure that there are no holes in your scoring function, because if there is a hole that it kind of generate, then it will generate molecules that so to say exploit that hole in your shape or in your QSR model. That might lead to unrealistic molecules. So this is a short video I will show. It's a toy example. So that's the goal here, is to generate molecules that is similar but not identical to celecoxib. And if you start the video, you can see it samples in the chemical space. On the bottom, you see the scoring function. It's flat, so it's sample around, but doesn't find anything that is remotely similar. But then suddenly it starts to find some molecules that's a little bit similar, and then for reinforcement learning, it generates more and more similar compounds until you have reached the threshold of 0.7 that we set in the input. And of course, this is a video. In reality, the generation of molecules is much quicker than this, but it's illustrates the principle how you can actually, through sampling, sample the whole chemical space, and then for reinforcement learning, hone in to the chemical space that is of most interest. And we have a platform called Reinvent, where we have plugged basically all the components together, the generation of molecules, then the sampling, and the scoring. And the scoring could be QSR models, docking, shape, and so on. And then you have a reinforcement learning loop, so you can really move around in the chemical space. You through that can find new scaffolds and so on. And then we combine it to the left. There's AI synthesis prediction tool, and then as much as we can to the right with the iLab, the chemist automation platform. So this is a picture of the automation platform that's built here over in Gothenburg, and this is a more description of it, that it's basically from the design. You have a building block store, and then you have the synthesis robots, either flow or batch, and batch usually mean 96 well chemistry. Purification, you do the analysis, you do the evaporation, then you can do the quantification, or you can do the risk suspension, and then do the testing. So currently we do both uses for parallel chemistry, they use it for high throughput experimentation. But if it's a favorable type of assay that is easy to also include, we can also do closed loop optimization. So one big project we have in automation is basically how can we make more chemistry amenable to automation. So we've analyzed our electronic lab notebook going back to 2003 to find the most common chemical reactions to optimize. We have seen which they are, and then we're currently working on optimizing for the platform the 60 most common chemical transformations. And so what can we do today on the platform? So we can basically do high throughput chemistry arrays up to 96 compounds. Could be parallel library or high throughut experimentation. We have cycle times from synthesis until you have a screening ready stock solution for 1 to 4 hours. And currently we can do 20 plus different chemistry transformation. What we still need to develop there is the multi-step synthesis with intermediate purification. I don't think anyone is actually doing that routine in the pharma industry at the moment, but that's definitely an ambition that we have. We also need to work on automating supply of chemical building blocks. And then, of course, when we have everything in place, also to do the full integration with the Reinvent, the chemistry AI platform. I've spent a few minutes also to talk about an exciting project called MELLODDY. It's, I think, a very innovative way to how you can improve your QSAR, your chemogenomics modeling. So it's an IMI2 project with 10 pharma companies and a set of public partners that is working together with privacy-preserving machine learning. So basically what we're doing there is that we are using a feed-forward network, and what is very important is that all sensitive data, saved data, and let's say chemical structures, they are never leaving a partner's AWS instance. So it's only the layers in the feed-forward network, not the most sensitive last layers, but the earlier layer that are then mixed and trained. And that's done according to a scheme through a ledger. So we can really track which models are from a pharma that is actually trained with which model from another pharma to improve the accuracy of the model. So it's done in the cloud, AWS, through a ledger that decide actually automatically which models that will be trained with each other. And the technology stack, so the algorithm is developed by KU in Belgium. So it's multitask learning feed-forward network. We have the application layer, which is from a French company called ORCHESTRATION. It's a traceable federated learning platform. And since we run it on the cloud, we need, let's say, an orchestration that orchestrates the computations. And that's the Kubernetes, which I think is a standard tool to do that, and it's developed by a company called Kubernetes. And then on the bottom, the infrastructure is run on Amazon Web Service, where each pharma company have their own instance, and where no sensitive data is leaving that company's instance. And we just finished year one run, and that's was a proof of principle that the platform is functional. We also did a vigorous security audit to make sure it's safe for all the partners, and all partners approved the audit. We started to run with the 10 pharma partners, and we did this year mainly used dose response data with over 70 million data points. And we can show that it works. We didn't optimize the machine learning. That's more for the second year. This year was a proof of principle. We have a platform that works. And then year two is really the goal is to understand how much we can improve our models when we train the multi-pharma versus single pharma. As a conclusion, I think I'm running out of time, so there's some questions. So to say, the first is we're really working with scaling AI and automation for the whole, so to say, the portfolio project we have, and that's of course include more challenging project with low data volume. Finding affinity and also solubility are still bottlenecks, and finding affinity, we try to approach both with free energy perturbation, but also innovative way of workings like MELLODDY. Not so much coming to that a big challenge for us is to read all the scientific literature. Everything that comes out is really publishing a lot, and actually to assess the progress, to understand which methods are coming out of really progress in the field. And of course, if you want to do a change in a company to apply more machine learning AI for drug design, it's also an educational, culture and logistical challenge. And also, of course, the bar is set high to transform drug design. I think I always get that question if you think we'll do that. And the jury is still out there. I think we have shown a lot of impact already, but to transform a whole, so to say, way of working, we are still not there. But of course, that's the ambition. To put it in perspective, I think the last time it was really transformed, it was in the 90s, when the DMPK department were installing or so to say started in the big pharma companies. I'll say thanks a lot. There's a lot of people who've been involved, as you can see, particularly the team to the left. Also, my line manager, Gary Parado, which is actually have just left AstraZeneca, has been very influential. Also for application, a lot of support for the 3 disease areas, respiratory, oncology, and cardiovascular, a lot of academic collaborators, and also funding, public funding for PhD students, where we have together with the University Weekend, which was an EU-funded ITN, and also funding within Sweden from the Wallenberg Foundation to train PhD students in Sweden in AI and machine learning. And with that, I say thank you, and thanks for listening, and I'm ready to take a few questions.
Thank you very much, Gyula. We have time for a couple of questions. So the first one is, can you optimize towards multiple objectives simultaneously with the generative neural network?
Answer is yes, that we do regularly. I mean, in the end, drug design, per definition, is multi-parametric. Clearly, it's if you put in more and more parameters that we like to optimize, particularly if they're a little bit not going the same direction, you want to have both solubility, high solubility and high permeability, then something in the end, something need to give in the optimization procedures. And that could be, for instance, synthetic accessibility, that the proposed molecules is not 3 to 4 steps, but maybe 6 to 8 steps.
We have another question from an anonymous attendee who asks, why are solubility predictions a major bottleneck?
I think first, of course, it's Solubility is a bit tricky to measure as well. So if you need to see from the training set, you have both, of course, thermodynamic solubility and what's called kinetic solubility from a more amorphous state. I think it's really with QSCR models to predict the crystal structure, the energies in the crystal structure that is the bottleneck, which makes it a bit more difficult than to predict, say, log D, which is the equilibrium between two liquid phases.